Good morning. My name is Justin George, and today I'm going to be talking about my research project, which focused on the toxicological effects of bisphenol S on Planarian regeneration. In recent years, plastic has seen a massive increase in production, with annual plastic production reaching 507 million tons a year. But these plastics not only contribute to the global plastic pollution problem, but they also pollute our ecosystems with harmful chemicals that impact the organisms living within them. One category of these commonly used plastic chemicals are endocrine disrupting chemicals, or EDCs. And these EDCs have the ability to mimic and block hormones within an organism and disrupt hormone signaling, which can cause detrimental problems. One of these EDCs is bisphenol S, or BPS, which is the chemical that I decided to focus my research on. In recent years, BPS has grown in popularity as it has begun being used as a substitute for BPA. You'll see many products marketed as BPA free, but oftentimes these products, rather than having BPA, have BPS. But the thing is, uh, due to the structural similarities of BPA and BPS, it calls into question whether BPS is truly a safer alternative. To study the effects of BPS, I decided to use the model organism Planaria. Planaria have the unique pluripotent stem cells known as neoblasts. These neoblasts are able to differentiate into various types of cells during tissue regeneration and replace lost parts. And when these planaria are amputated, a mass of these stem cells aggregate at the wound site, creating a blastema. And this blastema is important because it is uh, an easy method to quantify the regeneration rate of planaria. Now, onto my hypothesis. I hypothesized that there would be a dose-dependent relationship between the concentration of BPS and the blastema regeneration, as in that higher concentrations of BPS would cause a more significant delay in blastema regeneration. I also hypothesized that the control groups used, both the negative control and the vehicle control, would not have a significant impact on the regeneration rates. In order to conduct this experiment, I first started by creating two treatment groups, a 500 micromolar BPS solution and a 100 micromolar BPS solution, in order to simulate high and low concentration environments. I then went on to create two control groups, a vehicle control group with ethanol, since ethanol was used to dissolve BPS, and a negative control group of just plain water. After this, planaria were amputated transversely, and the inferior ends were placed into each of the treatment groups. Over the course of 2 weeks, the regeneration of these planaria were observed and measured. Images of these planaria were taken every other day and analyzed through ImageJ analysis in order to measure the blastema growth rate. Also, qualitatively, I looked for the regeneration of the photoreceptors, which are similar to the eyes of the planaria, and noted the days in which a photoreceptor formation was complete. The results were just as hypothesized. As seen in the graph, the 500 micromolar concentration, or the highest concentration, which was the blue line, had the slowest regeneration rate, but then the 100 micromolar solution, or the red line, had a higher regeneration rate, and then finally, the vehicle and negative control groups, the green and yellow lines, had the normal regeneration rates. This showed that there was a dose-dependent relationship, with the highest concentration having the slowest regeneration, and the lowest concentration having a greater regeneration rate. By the negative control group and the vehicle control group having similar regeneration rates, it shows that the ethanol did not have a significant impact, and that the results seen in the two treatment groups were solely due to the BPS. In conclusion, these results highlight that BPS isn't as harmless as was previously thought, but rather that it may have significant biological effects on the organisms exposed to it. This raises significant safety concerns due to its widespread use and highlights the need for stricter policies on BPS usage. BPS is still heavily understudied and must be studied more to understand the full effects of BPS. Future research can be conducted in many fields, whether it's diving deeper into long-term and multi-generational effects, like effects on behavior, lifespan, and heritable effects, or by studying the many other BPA substitutes that have begun to emerge in the industry, such as BPF, BPAF, BPAP, and many others. Furthermore, it's also important to expand the range of BPS concentrations tested. In my experiment, due to the limited resources and time that I had, was only able to test two different concentrations. However, by testing uh, a wider variety of concentrations, it would allow to narrow down the exact range at which toxic effects of PPS begin to manifest. Here are just a few of the many resources that helped inform me throughout my research. Thank you so much for your time, and feel free to ask me any questions that you may have.